Hello friends, welcome to With My Plan. Last class we understood the gibberellin signaling in plants. There we understood what is happening in presence of gibberellin and what is happening in the absence of gibberellin. Today we will deal with the topic gibberellin signaling in seed germination. It means we are going to deal with the signaling process of gibberellin in germinating seeds. Okay, so let's start. So this is the diagram of signaling pathway here. Let's start with the points. I have put the numbers in the diagram 1 2 3 like so. Accordingly the points are mentioned here. Okay, so in the first point we can see initial step involves the gibberellic acid from the embryo binding to the cell surface receptor, and these things are happening in the aileron layer. Okay, we have already read this. Aileron layer is having is unicellular layer. Those cells are basically rich in protein, that's why the name is aileron layer. So in the aileron layer what is happening, GA is coming from your embryo from through endosperm, scutellum and endosperm, and it is reaching to the aileron layer. Aileron layer cell wall and cell membrane, it is going through, and in the cell membrane there is receptor of GA which is receiving the GA molecule. So in the second step what is happening, the GA receptor complex at the cell surface engages with heterotrimeric G protein. So in the membrane itself, there is G protein which is activated by the receptor, or the active receptor of GA after receiving the GA molecule. So this G protein, what is happening, it has three subunits: alpha subunit, beta subunit and gamma subunit, and also it is attached with GTP. So on activation, whole part will be dissociated, and it will form CGMP, which will act as a signal transduction or signaling intermediate for further signaling. So in the third step, the pathway independent of calcium, which includes CGMP, leads to the activation of signaling molecule. So what is happening there, after activation of G protein, there will be two pathway. One is calcium dependent and another is calcium independent. So calcium independent will basically involve the signaling molecule, signaling pathways, and calcium independent will involve the release of or the secretion of the alpha amylase enzymes. So both have important role. Let's understand first the calcium independent pathway, which is getting activated with the help of the CGMP, which is coming from this G protein. Okay, so the activated signaling molecule, or signaling intermediates basically enter into the nucleus. In this through this signaling calcium independent pathway, this activated GA signaling intermediate will enter into the nucleus. This is the nucleus. So after entering into the nucleus, what is happening, there is it attaches to the DELA protein. DELA is basically a repressor of gibberellin within the nucleus. So within the nucleus, this GA signaling intermediate will attach with the DELA protein, and after attaching to the DELA protein, it will start the degradation process of the repressor DELA. So what is happening, the inactivation of DELA repressor enables the expression of the MYB gene. The degradation of DELA repressor occurs when they are bound to the GA signaling intermediates. And next, what is happening, the inactivation of DELA repressor enable the expression of MYB gene along with the other genes to progress through the transcription processing and translation. So what is happening, this DELA protein is getting degraded by the GA signaling intermediate, and apart from this it will attach with the F-box protein, and there will be degradation of DELA through ubiquitination, where 26S proteasome is involved. Okay, so through this repressor will be degraded. Now this signaling intermediate will involve, it will attach to the promoter region of GAMYB gene, and it will produce the mRNA, or synthesize the mRNA of GAMYB. And that mRNA will come to the cytoplasm and convert it into the transcription factor. Basically, this transcription factor will again used as a will attach to the promoter region of alpha amylase, which will again express the gene of alpha amylase. So what is happening next, the recently synthesized MYB protein subsequently enters into the nucleus and attaches to the promoter gene responsible for the alpha amylase and other hydrolytic enzymes. Okay, the activation of transcription occurs for alpha amylase and other hydrolytic enzymes. Alpha amylase and other hydrolysases are produced through rough endoplasmic reticulum, and proteins are released through the Golgi apparatus. Now, GA stimulation is necessary for the secretory pathway through a signaling transduction pathway that depends on the calcium calmodulin. So what is happening, as I told here, ki GAMYB mRNA is producing, or transcription of protein is happening here, translation of protein is happening here, and that protein is coming to cytoplasm and entering again to the nucleus, being a transcription factor, and that transcription factor will attach to the promoter region of alpha amylase, which will again help in transcription and translation of alpha amylase. That alpha amylase will go to the endoplasmic reticulum and then to the Golgi body, through which it can be secreted. First it will come in packets, or through membrane bound vesicles, and lastly it will exit, or secreted through the membrane to the endosperm, and that those hydrolytic enzymes including alpha amylase will simplify the, or degrade the complex food material into the simplest form, so that the plant can easily use, or the embryo can easily use it for initiation of the plumule and radicle in the seedling. So this is all about the signaling of um, GA in seed germination. So that is all. I guess you understood everything. If you are having any doubt, please comment below. I will try to solve it. Thank you.